Okay, thank you, Susan. Good afternoon, everyone. My name is Pedro. I'm a PhD student here at Birmingham, and today I'll be talking to you about our work on the evolution of body size in crocs. So, well, when we look at these very large animals such as whales and sauropods, it's easy to understand why people engage studying size in these animals. So, and in fact, when we look at the study of size in tetrapods, most of the work has been done mainly on mammals and dinosaurs. And that's not only because of some of their species were very, very big, but also because of their very rich fossil record and their huge diversity. Okay, but when we look at Crocodylomorpha, as Phil just said, we also see a very rich fossil record and also very huge diversity and morphological disparity. And for our work, we also see a wide range of sizes. So we have very large animals and very small animals as well. And so because of this, we have a good scenario for studying body size evolution in this group, in Crocodylomorpha, and that's why we started doing this. So there are many ways of doing this, of body sizing in lineages, and more recently people started doing this in phylogenetic context, and using, for example, evolutionary models. And for this, basically, what we need is, of course, a data set of size. We need a time-calibrated phylogeny, and of course, evolutionary models. So let's go through these items for our study. As you all know, it's very rare to find completely preserved skeletons of tetrapods, despite of this seen in Jurassic Park. And more often we find isolated bones, such as vertebrae or long bones, but in crocs we also very frequently find articulated skulls. And that's why we decided to use a cranial measurement as a proxy for total body size. And not only one cranial measurement, but two, because with this we could increase our sample size and also test if these different measurements could have an influence in our results. Okay, going further, as the phylogeny, we decided to use an updated version of Super3 published in 2015 because it's very comprehensive and we wanted this. But we also used two alternative topologies in which we changed the position of Thalattosuchia, the marine crocs, because of their very controversial phylogenetic positions. And finally, the evolutionary models. As you know, evolutionary models assume different types of rates and modes of evolution. For example, in Brownian motion, it assumes that the evolution of a given trait in a given lineage evolves randomly. Many other evolutionary models that assume different types, different rates and modes of evolution. And in our case here today, an important one is, it was supposed to be highlighted, the OU model. So the one important for us today is the OU model. It's a non-uniform model, and the important thing about the OU model is that the evolution of a trait occurs within an evolutionary regime. So evolution is constrained within this evolutionary regime. So it has, so the trait is evolving and the lineage is evolving. Their trait is attracted towards a trait optimum, and it's possible to calculate this trait optimum and also the strength of attraction toward this. And more recently, people have developed a method and also a new evolutionary model that is called the surface model. And the new thing about this is that it allows multiple trait optima to occur. So in other words, it allows multiple regimes or shifting regimes to occur. And it's also possible to identify convergent regimes, which means that we can identify convergent evolution happening in a clade, in a lineage, for example. Okay, just summarizing all this stuff I just said, for our model fitting analysis, we had two data sets, two time calibration methods, because this can have influence in our model fitting results. We had three topologies, three phylogenies, and we randomly resolved 10 times each tree. And this gave us, in the end, 120 analyses, and we fitted each one of these 5 models to all of them. I won't be showing you all the analysis because, as I just said, there's a lot of stuff. But believe me, all of them showed similar results. So what we see is that the surface model offers the best fit in all of them, by far. And this means that the surface model is the best explanation for the evolution of body size in Crocodylomorpha. And because of these very significant results, better results than the other models, we decided to give a closer look in the surface model using the R package surface. And this is the sort of output tree or output result you get from surface. And this is just for one tree, not all of them, because I will show all of them for you. The circles represent different regimes, and the colors, same colors, represent convergent regimes. So what we also get from surface is a lot of important information, but here for us, for example, in this tree we had 24 regimes and 8 convergent regimes represented by 8 colors in this tree. And what is also possible to get is the actual trait values, the trait optimum values for each one of these 8 convergent regimes in this tree. And of course, I'm talking about just a cranial measurement, not the whole body of the animal. That's why these values are in centimeters. Okay, but if we want to be sure that we are looking at real patterns, not just something that could happen in one tree, you need to look and try to find general patterns in all the trees, right? So what the first obvious thing we can see, for example, in this example tree, is that there are many regime shifts. This means that although body size evolution in crocs is constrained within regimes, it also shows that there's a lot of different things going on there, which makes sense when we think that we are talking about a very diverse group that had a very long evolutionary history. And yeah, this is represented by these many regime shifts. And there's also some other important aspect that we could identify in all of the trees. For example, most basal crocs, they were very small in general, and they didn't experience any regime shift there in the base of the tree. That is represented here by this red color clades species. And they stayed under this same regime until the early late Cretaceous, and they didn't change much, so they were generally smaller than the others. Another important thing that we see that in all trees is that the 2 main lineages of Crocodylomorpha, Neosuca and Notosuca, they show a lot of regime shifts, which is expected because they're very diverse. Most crocs are included in these 2 groups. And also, when we look at the crown group, Crocodilia, we see right at the base of the group, we see a regime shift towards relatively larger sizes. And 2 of the main lineages of Crocodilia are still under the same regime until today. So this might be significant for the success until today of these animals. Okay, so from the surface, we had the idea of a very complex evolutionary landscape in crocs, which makes sense when you compare with they're very diverse because they're very diverse. But trying to understand which factors could be driving this evolution, we did some exploratory analysis comparing our size data to other environmental factors. And initially, we did some correlation tests, temperature and latitude. And we did this using our 2 data sets that we had and using different sources of information for temperature, for example, and comparing this in time series, comparing temperature with maximum size, mean size within this time being. And after a set of analysis, a lot of analysis, we didn't find any significant correlation. This might mean something like that just our data is not correlated with this temperature data that we tested, But it also can mean that temperature was not actually driving the evolution of body size in crocs. And when we looked at latitude, we did find some significant correlation, but it's not very strong. It's a kind of weak correlation. So in the end, could also be a problem with our data, but it could also be because such factors, when we compare this with our data size, they cannot isolated explain the very complex evolution of body size in crocs. And in fact, what we could also do is trying to test this with other environmental factors, for example, using different sources of information for temperature and doing phylogenetic correlation corrections for this analysis. But so far, we're still doing this. So just the most important conclusions. First important thing was that the surface model offers the best explanation for the body size evolution in crocs. And this means that crocs evolved within, body size evolution of crocs was constrained within regimes, but as it also exhibits a lot of regimes, multiple shifting regimes, it's compatible with their very rich evolutionary history. And so far, we didn't find any support or strong support for correlation between size and these environmental factors that we tested, but this could change in the future with new tests. And I would like to thank all these funding agencies, people, and collections. That's all.